afternoon. I want to talk about emerging viruses to build upon what we talked about last time. What is an emerging virus? A causative agent of a new or previously unrecognized infection. The term became popular. In fact, it was coined in the 1990s, but emerging viruses is nothing new. It's just that we recognize this concept. They have always been emerging. Before there was life on the planet, viruses emerged, and they have so ever since. For most of the history of the Earth, there was no one around to watch them emerge. And only lately have we gotten good at detecting emerging viruses. Now, as far as human viruses go, I think since the rise of agriculture about 11,000 years ago, that gave us the ability to start congregating in big cities with thousands and thousands of people. And we had our animals for the agriculture that would sustain human populations. And so we began to get viruses from the animals, and we transmitted viruses more readily amongst each other. An emerging virus is one with an expanded host range, with an increase in disease that was not previously obvious. That's one kind of emerging virus. In other words, it's a virus that we know has been around, but suddenly it's doing something different. Great example, Zika virus, which we'll talk about today. It can also be transmission of a virus from a wild or domesticated animal to humans. This is what we call a zoonosis or a zoonotic infection, a virus coming from an animal, a non-human animal, into a human. And so that can be a brand new virus that we've never seen before, like Ebolavirus or HIV. And again, these viruses have been around way longer than we've been detecting them. So, Ebolavirus started infecting humans, as far as we know, in the 70s, but probably infected us long before. And that virus has been around for ages. We just didn't know it existed. Sometimes cross-species infection establishes a new virus in the population, like SIV moving from chimps to humans. We'll devote an entire lecture next time talking about that jump. And sometimes these cross-species infections are not sustainable. So Ebola and Marburg, going from bats to humans, is not sustained in people. Eventually, we're able to curtail these outbreaks. And the next one is always a brand new jump of the virus from animals to people. So sometimes these zoonotic infections get adapted to people and they become human viruses like HIV, and often they do not. We'll talk about this more today. So the human-animal interface is extremely important for emerging infections. The world has way more animals than humans on it, and they're the source of many of the infections we get. So here's a pie chart showing you the number of viruses in terms of genera in a number of different categories. In blue, 32 are what we call adapted pathogens. These are viruses that we got from animals. They infected Homo sapiens at some point in our history, and they become well adapted so that they're now human viruses, like the most recent one being SIV going into humans and becoming HIV. So HIV would be an adapted pathogen. Then we have zoonotic pathogens, 37 of them in red. That means that every time there's an outbreak in people, it's a brand new transmission from animals to humans. And that includes Ebolaviruses that we'll talk about today, and some others as well. Obviously, 37 other genera. Heirloom pathogens are the orange, 16 heirloom pathogens, less than Homo species. All right, so there are lots of Homo species, or 7 or so Homo species. These are viruses that we inherited from before Homo sapiens. All our ancestors, greater than Homo species, would be all the different Homo species that passed them to us. So 2 different kinds of heirloom pathogens that we've had for a while, either from the ancestors of Homo species or from different Homo species as well. But eventually all coming from animals at some point or another. And of course, whenever there are brand new viruses that we haven't seen before, this is a relatively recent phenomenon. I would argue that it was first recognized in the 60s with Lassa virus emerging in Africa, and that book Fever describes that, as I've told you before, a really good one if you can find it. But of course, when Ebola emerged, it's on the front pages of everything, and here's Ebola, of course. And of course, the press loves it because, as I said before, they love drama and tragedy. Why? Now, recently, we have seemed to have more novel pathogens infecting us, and that's probably true. And there are a number of factors that contribute to that. And some of them are shown here on this slide. And they, of course, include the ability for humans to travel all around the world, expanding cities, and lots of poor people in many cities that have conditions that foster the spread of infections. Deforestation is a big part of it as well. People that go in and deforest are susceptible to infections. We alter ecosystems all the time, and that changes the interactions between species, and we change the environment extensively. And of course, at the middle of all of this is the ability of viruses to evolve, as we talked about last time. All the different ways they can do that. They have huge potential, and we simply give viruses more means to infect us. Perhaps the biggest driver, though, is the huge human population growth and all the changes that causes on the planet, which is really summarized beautifully in this graph of human population numbers in billions here on the y-axis. And you can see for so many years we were just a tiny, tiny species. And then all of a sudden, look at this explosion in billions and billions of humans, occupying almost every part of the planet and encountering more viruses, of course. So it's not surprising that we have more zoonotic infections. The world is full of animals of all sorts, and each of them has its own collection of viruses. So here, just to show you an example, is the Amazon North region of Brazil. Right here you have it here, Brazil, Amazon North, and right there in green. And these are the different areas, and these in print that you can't read, I understand, are the names of a variety of viruses that are known to be present here. 183, this is a few years old now, so it's probably out of date. Some of these have actually been isolated. Others, there's other evidence that they are circulating. But you can see many, many viruses. Some of them are arthropod-borne, vector-borne, mosquitoes, for example. Many of them infecting vertebrates. So all of these, as we go into the Amazon and deforest it and destroy it, we encounter animals with these viruses, and they have the potential to infect us. So there's no shortage of candidates for future zoonotic infections. The only question is when it will happen. So here's a table of a number of viruses which we have seen either newly emerged in recent years or have expanded in their ability to infect globally. And they include some viruses we have talked about, but the asterisk one we're going to talk about today, Ebolavirus, for example, the driver of emergence, human contact with natural host bushmeat trade, Hendra virus, a paramyxovirus we'll talk about, which travels from bats to horses to stable workers, a virus we didn't know about up until a number of years ago. HIV we'll talk about next time, probably crossed over from chimps to humans via the bushmeat trade. Nipah virus, from bats to pigs to humans. And the specifics we'll talk about today. Nombre is a virus that emerged due to changes in weather, as well as human and rodent contact. We've talked about the introduction of West Nile virus to the US. You introduce a virus into a country where it's never been before, no one has immunity, and it makes a huge outbreak. And finally, Zika virus, spread via mosquitoes and recently emerged as well. So obviously evolution has a big role to play in emerging viruses because evolution leads to biodiversity, quasi-species. Remember, every virus out there isn't one sequence. It's a mixture of thousands of sequences. And all those have to do is find a new host, and one of those will take off and make its own quasi-species. So this allows viruses to adapt to new hosts and environments using the mechanisms we talked about last time, variation, and selection. Now, when we talk about the interactions of hosts and viruses, we'll put them in a framework. We'll put the interactions in a framework so that we can understand what happens in an emerging infection. So here's the diagram I like to use. It has 4 different patterns of host and virus infection. So the first one is a stable one in the green box. And this is a stable virus-host interaction that's been going on a long time, maintains virus in the ecosystem like measles virus infecting humans. It's been adapted to humans for thousands of years. It infects just them, and that's a stable virus-host interaction. Then we have evolving. This is what we get when the virus is passed to a naive population. In other words, a population that's never seen the virus before. It can be the same host. It could be humans, so it could be the introduction of Zika into a naive population, the introduction of West Nile into a naive population, or it can be the introduction of a virus from an animal to a human. Maybe you're thinking, do we ever give viruses to animals? And the answer is yes. Some people call that a reverse zoonosis. There are examples of people doing that in various situations, mainly in zoos, because that's where most people get pretty close to animals. But there's a recent example in Africa where contact between humans and wild animals may have led to infection as well. Then we have dead-end infections, one-way passage to different species, the red one. And this is mostly what happens out there. We encounter viruses all the time and they don't go anywhere. Sometimes they cause disease in us. So I would say Ebola is a dead-end infection because the virus never sustains transmission, never becomes a human virus. We can suppress the outbreaks, and the next one has to come from another zoonotic infection. So that's a dead end. And finally, resistant host, replication is blocked. So we probably take up viruses all the time. For example, you go to the supermarket and you buy cabbage or coleslaw. Depending on what time of year, it is covered with insect viruses. People have studied this. Thousands and thousands of viruses per gram of coleslaw or cabbage. It comes from insects that crawl over the cabbage in the field, and they're excreting these viruses, and washing doesn't get rid of them. You eat them, they pass through your intestine. If I sampled any of you, if you happen to have eaten cabbage, I would find some insect viruses in you, but they don't replicate in you. You're a resistant host. And there are many, many other examples of that. Now, in this scheme, these arrows show directions between the various situations. The red ones are the major pathways. So, for example, we can have a stable virus-host relationship. Let's say Ebola virus in bats. That's been in them for years. In fact, the bats are probably okay with that infection. And then it gets introduced into people. It becomes evolving because it's rapidly changing. The quasi-species is trying to be selected to be adapted. And so that's where the evolving infections come from. And from stable relationships also come dead-end infections and as well resistant infections. So many of these can trade amongst each other, but the main ones are in the red arrows. So let's talk about some of these in some more detail. Stable host-virus interactions, both parts of the equation, virus and host, they both survive. And some of these are long-term or permanent. Humans with measles, herpes simplex virus, they've been in us for many, many years, as well as other species as well, cytomegaloviruses, smallpox. And sometimes more than one species is involved. Influenza viruses, flaviviruses, and togaviruses all can not only infect us, but other species as well, and can maintain stable relationships in multiple species. Evolving, again, is an example of a zoonotic infection where a virus changes hosts. The interaction is unstable and unpredictable. Sometimes nothing happens. There's no symptoms of infection. Sometimes the individuals die. Some good examples that illustrate this, there are just many, many, many examples of it, not just Ebola, but introduction of smallpox and measles to natives of the Americas by old world colonists and slave traders long, long time ago. Some of these viruses evolved in the Middle East and Europe. They weren't in the New World, but they were brought there by people who traveled, and because These were nasty human viruses to begin with, and the population in the New World had no immunity whatsoever. It wiped them out. West Nile in the Western Hemisphere, same thing. No immunity in the US. The virus could spread extensively. And then last time we talked about the introduction of rabbits into Australia, but more precisely the attempt to wipe them out with a virus. So here we have a naive population of rabbits. We put a virus in that's lethal to them, and in the first year it kills many of them. So those are all examples of evolving host-virus interactions. A dead-end infection, often what happens when you have a zoonotic or cross-species infection, you don't get sustained transmission from a new infected host to others of the same species. So in the case of Ebola virus, which we'll talk about more later, we think the reservoir is bats, but we're not absolutely sure, but there's some evidence for that. And then it's in a stable relationship in bats, and then cross-species infections occur in humans, in chimps, in gorillas, in which it is a dead end. It doesn't seem to spread extensively to others, and it can be lethal. H5N1, avian H5N1 influenza virus, a bird virus that is circulating in bird populations in many parts of the world, can occasionally infect people. It's a dead-end infection. It does not transmit among people. And people are very worried that it may evolve the ability to transmit, or I should say a mutant will emerge that in the quasi-species that's able to be transmitted, but that hasn't happened in the 50 years or so that we've been watching it. So these kinds of dead ends contribute little to the spread of a natural infection, because the natural infection is in a different species. It's a stable interaction from host to host, and the dead ends tend to die out. So here are some examples of these sorts of life cycles. This is an arbovirus that engages in a number of stable host-virus interactions. We have wild birds, amongst which the virus is transmitted by mosquitoes. Mosquitoes do like to bite birds. They don't just bite us. There are many species out there that bite birds. So the virus is transmitted in a stable host-virus relationship between the birds. These birds, if bitten by a different mosquito, can bring the virus to other hosts and other stable relationships as well. These mosquitoes can also bite chickens, and they can spread virus to them. The mosquitoes, of course, can bite other hosts as well. When these arboviruses, arthropod-borne virus of a number of different families, when they bite horses or humans, they transmit the virus, but these are dead-end hosts because we don't spread it amongst each other, nor do horses, and mosquitoes don't pick it up from humans and horses and spread it to other humans and horses. Apparently, there's not enough viremia or other factors involved. So we are accidental hosts. The mosquitoes will bite whoever's around, and if they have viruses in them, they'll deliver it, but that will be the dead end. These humans, these horses, may not get any disease. They may get mild disease. They may die. It's not relevant to whether it's a dead end or not. The fact is that it's not transmitted effectively. Here's another example, European tick-borne encephalitis virus. So this is a different vector. It happens to be a tick, which spreads the virus in a stable host-virus relationship amongst rodents. The ticks also transmit the virus vertically through the eggs to other ticks, to ticks that are born from the mother. They can be immediately infected and spread it to rodents as well. The ticks can then bite people. You go walking in the woods, you get ticks on you, they bite you, transmit the virus, you may get sick, you may die. You're a dead-end host because you're not going to spread it to anyone else. Ticks are not going to pick it up from you, and you're not spreading it by any other means. There's no aerosol transmission, etc. Sometimes the ticks bite goats. We drink goat milk and we get the virus as well, and that's also a dead-end infection. So here's another example of the different host-virus relationships. These are stable host-virus relationships among the Flaviviruses that are known. There are lots of Flaviviruses, but these are the ones that are known to be human pathogens. And they're divided into three groups here, which are different in the amplifying host and the vector. So the top three, West Nile virus, Japanese encephalitis, St. Louis encephalitis virus. These are all amplified in birds, so the viruses grow in birds. They're picked up from mosquitoes and transmitted among the birds. Occasionally they will infect people, and there can be outbreaks among people transmitted by mosquitoes as well. Obviously, West Nile is transmitted very well from person to person. Here we have a group of viruses in orange where the amplifying hosts are primates, and it's transmitted by a different kind of mosquito, Aedes mosquitoes as opposed to culicine mosquitoes with the top three. And here we have our 4 serotypes of dengue, Zika virus, yellow fever virus, and these are all transmittable among humans by the vector. So they don't have to go back to the reservoir host each time. And they're all in stable host-virus relationships with humans as well. Here we have an interesting one, Spondwaini. Anybody hear of Spondwaini? But you might someday, maybe the next Zika, who knows? It's out there, it causes infections and disease. Not on anyone's radar, except for a few. Maybe one day it'll emerge. And finally, the last one is tick-borne encephalitis, which is the tick-borne virus, rodents and humans. In general, in order for a virus to establish itself to become an emerging infection, there are 2 steps. First, it has to be introduced into the new species. It has to be introduced into us, obviously. And then it has to become established and disseminate. And those are pretty high barriers, as you'll see. We have lots of ways to meet new viruses. As I alluded to earlier, our society changes constantly. Not only does population grow, but we do lots of things. Some of them are shown on this slide that allow us to meet new viruses, making dams, changing the way water flows, irrigation, deforestation, making wildlife change, wildlife parks, long-distance transport. Then, of course, we have air travel, urbanization, daycare centers, big one. A lot of outbreaks in kids that get brought home. Hot tubs. Why is hot tubs here? Because we don't chlorinate them properly and they become contaminated and we can spread virus infections that way. Air conditioning, the aerosolation of fluids that are contaminated. Use tires to spread mosquitoes, as we've talked about before. And then we have medical procedures like blood transfusions, where we've transmitted very many viruses over the years until we've discovered them. Xenotransplantation. This is using animal parts to replace parts of people, typically like a heart valve. You can put in a heart valve from a pig. Well, guess what? Those pigs have their own sets of viruses in them. And what are they going to do? People think about that all the time. And then, of course, societal changes with respect to drug abuse and sex has ended up spreading viruses in those ways as well. So we're very good at this. So we have many, many encounters with new viruses. Most of them we never even detect. As I said, they're happening on a daily basis. The insect viruses in your gut being an example. And even if a single host in the world is infected with a particular virus, it's never going to take off. It may never replicate properly into high enough levels. You may not encounter another person and so forth. Lots of reasons why these may not be transmitted. But we do know many examples of transmissions from animals. As I said, SIV from chimps to humans is one that did take off. And we'll talk about the factors that led to that next time. But we also encounter viruses from a variety of other species of horses, pigs, chickens, routinely. And many of them go nowhere. Occasionally, influenza virus will take off from one of those sources. And don't forget, it's not simply contact with bushmeat in the case of SIV or these other animals, but it can be viruses delivered by arthropods. Who knows what the mosquito that's biting you is delivering? Whenever I see a mosquito biting someone, I always think, what's in there and will anything happen? Most of the time, nothing will happen. But it only takes one in the right host to do it. And when you are exposed to new viruses, the viruses, of course, have to find susceptible and permissive cells in that host. And if they don't, that's the end of the line. Whether the virus is going to spread to other people depends on the population density, the health of that population. If there aren't many people around, you're not going to effectively spread the virus to anyone. But if the population is undernourished and, as a consequence, immunosuppressed, this could facilitate transmission to other hosts as well. But remember, in the end, viruses emerge only when you get serial transmissions from one host to another, to another, to another. So it has to get out of that first host to a second and a third and so forth, because we're not going to see it. Although nowadays we can see one or two unique infections here or there, and I'll give you an example of that today. Most of the time we don't notice it until it's quite extensive. I like to always bring up some examples of diseases of colonization, of exploration, because even though they're kind of artificial situations, they illustrate what happens when a virus comes into a naive population that's never seen it before. Now, in these cases, it's artificial because it happened at a certain time in our history when there was very little travel among different parts of the world. Then so we got isolated pathogens in different parts of the world. And then when people began to travel, we spread those. And that's when you get explosive epidemic spread, when the virus enters a naive population. Smallpox reached Europe from the Far East. So remember, it emerged possibly in a gerbil in the Far East in 710, then reached Europe because travel there was easy to do. There's no ocean in between, and caused lots of damage in Europe. And then, of course, it was brought to the New World, where in 1520, it spread from Hispaniola and killed 3.5 million Aztecs in 2 years. And that's why Cortez was able to conquer these individual societies, because of infectious diseases, not because he was such a good soldier, but most of the people were dying of smallpox, and his men were relatively resistant. They'd probably had it before, and they were the 70% or so that would survive after an infection. Now, you can imagine, though, that they brought some other viruses as well. Maybe they never took off in the native populations for whatever reason. Smallpox was one that happened to work. So let's go through a series of host-virus interactions. I would classify all of these as emerging at one time or another, and we'll learn some principles from them. And the first one is the emergence of epidemic polio at the start of the 20th century. So if you look in historical records from the Egyptians forward, here's a carving from thousands of years BC. It shows a priest, which looks like he has polio. He has a dropped foot and a withered leg, which are symptoms of polio. We don't actually know if he had polio, but you can find lots of examples of this over the years. In the 16, 17, and 1800s, you can see descriptions of cases which look like polio. Starting at about 1780, there are pretty good medical descriptions of kids with paralyzed limbs, but they're very sporadic. There was a case here, a case there. Only at the beginning of the 20th century did they start to occur in epidemic form, 10, 20, hundreds, thousands of cases of paralytic disease. All right, so this has been in a stable host-virus relationship with humans for many thousands of years. So what changed? Well, if it were today that this were happening, immediately, what would people say? What changed the pattern of polio infection? What would the press say? The virus mutated, right? Because I harp on all the time here. They would say, blame the virus mutating. Well, back then we didn't even know what the genome was. We didn't have any sequencing. Turned out that improved sanitation was the cause. It delayed transmission. So when you were born, within your first few months of life, you got infected with polio because poor sanitation, fecal oral transmission virus was everywhere. But you were okay mostly because your mother gave you antibodies against the virus. That protected you, but you built up your own immunity. So then we developed toilets, one of the biggest developments in modern history, the toilet and sewage, and we avoid infection for a couple of years. And your mother can't help you anymore, her antibodies are gone, and then you get polio. But more than that, you get big numbers of people who are susceptible, and the virus comes in, and boom, you have an outbreak of polio. That is what changed the epidemiology of polio, not the genome changing. I'm sure if we looked at the genome, we would see changes, right? Because viruses always mutate. And people, if this, as I said, if this were happening today, people would blame the genome. But this is a great example of how you should look at other places first. And so here are the numbers of polio cases. You can see one little outbreak at the end of the 1890s here. This is probably in Sweden, 32 cases or so. And then at the turn of the century, boom, Look at this. Lots and lots of cases till we reach our 20 to 30,000 a year. That's a very, very specific epidemiology. So again, the virus is present all these years. It changes its epidemiology because of delayed infection caused by sanitation. Bats have lots of viruses in them. Why this is so is very interesting. And there are some people who are trying to study bats. It's not easy to do. You can keep them in a cage, but that's not really what a bat does most of its life. It likes to fly around, and it flies its wings beat very quickly and generates a lot of energy. So bats have a very different immune system for dealing with all the oxidative radicals that are produced. And if you sit them in a cage all day, that doesn't quite duplicate that. But people would like to know why bats have so many virus infections and why they're okay with them, right? If you could figure out why a bat can survive, that might help us to figure out how to control some viral infections. Anyway, this is a flying fox right here. There's an acute. His wings are like this wide. He's huge. You don't like the looks of him? No. I think he's very cute. You can go sample these animals and catch them, and you can't kill them. It's not approved to kill them. But they have lots of viruses, and in fact, we found many new paramyxoviruses in these in recent years. Paramyxo, remember, negative strand envelope viruses like measles virus. And two of them, Nipah and Hendra viruses, I want to tell you about. These cause severe disease in animals and humans. Nipah and Hendra. So Nipah the first outbreak in 1998 in Malaysia. So again, this virus has probably been around for many, many years, but the conditions became right in 1998. It was an outbreak of respiratory and neurological disease on pig farms. Remember, now we're starting to grow pigs in greater and greater numbers for protein. It's a cheap, quick way to make protein. A pig matures in 8 months or so, and you get lots of protein for humans. So now we have pig farms with millions and millions of pigs on them. And so there was an outbreak on this particular pig farm, and the pig handlers, of course, they got infected. There were 100 human deaths. They had to kill all the pigs on this particular farm, 1 million pigs. And you know how they did it? They just dug a big hole and pushed all the pigs in. It's just a sad thing. I understand that they were infected, but you can find the videos of this on YouTube, where they just pushed them into a big pit. So they stopped that outbreak that way. But the way the virus began, so it turned out it was a new paramyxovirus, Nipah, which they subsequently found in fruit bats. And these animals are not affected by infections. They're infected continuously. They excrete it in their urine. And pig farmers, they plant mango trees near their pig pens, and the bats are nearby. They come to the mango trees, and they eat the mangoes, and they urinate while they're doing so, and the pigs come and they pick up mangoes from the ground that are contaminated, and they get infection. The pigs get infected, then they spread it to humans. So that's the source of this outbreak. So what changed here? Not only huge pig farming, but right next to the forest where the bats live. So you're moving in on the ecosystems that previously were pristine. Subsequently, it was shown in Bangladesh and India that humans could get infected by consuming date palm sap. So what people do in these areas is they put a sap collector on the palm trees and take out the sap, which is delicious and a delicacy. And many people were getting Nipah, and the common denominator was that they had drunk date palm sap. It turns out that at night, the bats urinate into the date palm sap because they're drinking it, and as they drink, they urinate, and they get virus in the date palm sap. So a very low-tech solution to stopping these outbreaks, just to cover the containers that you use to collect date palm sap, and that stops it. Infections do continue, however, because no matter how you tell people to do things, cover the containers, they won't. Some people won't, so there will be infections. And there's some human-to-human transmission, especially in cultures where the family prepares the body for burial. That ends up transmitting virus to other people because you have very close contact, obviously, with the infected body. It's very hard to get people to change their customary ways of burial. So that's Nipah virus. So to this day, we still see cases here and there of that from, again, transmission from bats to pigs to humans. It is not sustained human-to-human transmission, I would call this a dead-end virus. Hendra is the other one discovered in Australia, September 94. Here was an outbreak on a farm where there were racehorses, 14 racehorses, and one of the trainers that took care of them died. They identified Hendra virus, and what happened here is that, again, these farms were built right next to the habitat of the bats. So the virus, the bats come at night into the places where the horses are living. They contaminate their food. The horses acquire the virus from the bats. They get sick, and of course the trainers are very close to the horses, so they acquire the virus from the horses. And this continues to happen to this day. You can see outbreaks, small outbreaks of infection. Now, however, there's a vaccine which was made for horses. This is a very, very smart thing to do. You could never make a Hendra vaccine for people. There aren't enough cases globally. But racehorses are very costly animals, and you can easily convince the owners to vaccinate their horses, and that protects humans, right? That is called a one world health solution, because you have to recognize it's not just us on the planet, it's us and other animals, and viruses spread amongst us. So sometimes immunizing one will protect others as well. That was a vaccine developed in Australia. Those are two examples of building parts of our society next to habitats that harbor the bats. And now here's another one which is a little insidious because we didn't seem to do anything untoward in this case. This is an emergence caused by changing climate and animal populations. And the disease is hantavirus pulmonary syndrome, first noted in the Four Corners areas of New Mexico, 1993. So that's where the 4 states come together. There's a place called Four Corners, and in that area were these first cases of a very serious respiratory disease, mostly fatal. It was determined to be caused by a virus called Nombre virus. And this is a virus that's endemic in the deer mouse. The deer mouse is Paramiscus maniculatus. This is a mouse that is quite in many different places in the US. About 30% of any group of mice that you capture of this species are going to be positive for this virus. So if you like to camp or go into the woods, you have to be careful. Don't just pick up mice and play with them, okay? Because they could have hantavirus, and people have been infected. There are these wooden cabin type things which you can go and put your tent, your sleeping bag in, and they tend to be colonized by these mice, and they will produce virus that will infect you. There have been infections in upstate New York at campsites at Yosemite, California. Just 1 or 2 a year, not a lot, but they're typically fatal. So this virus was originally called Muerto Canyon virus because that's where the place was first isolated from, and the residents said, no way, you're naming a virus after our home. And the CDC went through a number of names, and nobody liked any of them, so they finally gave it Nombre, which of course means no name. So these are bunyaviruses. These are envelope negative strand viruses with segmented genomes in 3 segments, and the family is Bunyaviridae, so hantavirus is a genus within that family. And again, this had never been seen in the US, so what's going on? Why haven't we seen this? Well, turns out in 1992, there was a lot of rainfall, and this caused the piñon nuts to increase in number. So piñon nuts, people like to eat them. They're delicious. You cook with them. Wonderful stuff. But also mice like piñon nuts. So as the piñon nut population increased, so did the mouse population. And when you have more mice, the likelihood that they're going to go into your home increases. And the virus is excreted in feces, which then dries and it becomes aerosolized and can be inhaled. So humans are not the natural host. These mice are the natural host for the virus. But when you have human-mouse contact encouraged by these particular conditions, you get infection. And in retrospect, we could actually identify cases back to 1959. We just never thought they were this virus or knew it. We never detected it, never put this confluence of conditions together as was done in the 90s. Now, if you have mouse droppings in your apartment, do not just vacuum them up because that will create an aerosol. If there's virus in it, you're going to inhale it. You should spray them with bleach to inactivate the virus and wet it and then scoop it up. So the worst thing you can do is to vacuum dried mouse droppings. So add that to your list of things not to do that you have learned in this virology course. So here are the numbers of hantavirus pulmonary syndrome diseases as of January last year. There have been 690 diagnosed cases in the US since the 92 outbreak in these states. You can see they're mostly in the West. We've had a few in this area, a couple in New York State, Pennsylvania, et cetera. And you can see the range of the mouse here. This is Paramiscus maniculatus. So you can see that most of the US is positive except for the seaboard and the southeast, but it comes up to New York State here as well. So the range of this particular animal correlates with where the outbreaks are. But the virus has also been found in different kinds of mice. So this is a deer mouse, but we also find it in white-footed mice, the rice rat, and the cotton rat, which have different distributions throughout the US. So an interesting example of how changing climate causes a brand new emerging infection. It makes us recognize it, so we see it more often. And it emphasizes the fact that every animal out there has some kind of virus, multiple viruses that we are at risk to obtain under the right conditions. So here's an interesting example of a more recent one, which seems to be pretty rare. This is called Heartland virus disease. And this is first identified in 2012. It's a Flavivirus, which is a genus in the Bunyaviridae, just like the hantavirus is a genus in the Bunyaviridae, same kind of virus. So these were identified in 2 farmers in Missouri. So 2 separate cases of farmers came to the hospital with high fever and thrombocytopenia and a number of other indications. And smart doctors connected the 2. It turned out that both of them worked outside a lot, and they all had got lots of tick bites on a daily basis. They would come home in the evening and be full of ticks. And so people started to look for this. They found this virus in these individuals. They found 6 subsequent cases, which they think is also tick-borne here. But how long it's been infecting people, we don't know, and it's relatively rare. So here's an example where you can pick up a very rare infection. So I doubt that there are many more than these numbers of infection every year by being smart about your surveillance. And we have great tools now that we can use to pick up rare infections. All right, now let's spend the rest of the time, I want to talk about Ebola and Zika as examples of emerging infections. I want to talk about the conditions that lead to their emergence and what we have learned. So Ebola hemorrhagic fever was first recognized in 1976 in the Democratic Republic of the Congo. There were subsequent outbreaks, simultaneous outbreaks in DRC and Sudan, 318 cases and 284. The percentage is the case fatality in both. The number of deaths divided by the total number of confirmed cases, laboratory confirmed cases. So this is a very high fatality rate, as you can see. A similar virus had previously been identified in Germany in an outbreak from a primate colony. So primates that had been imported from Africa went into a primate colony in Marburg, Germany. It was an outbreak among the monkey handlers, which turned out to be a filovirus, just like Ebola, Marburg virus. And so that was actually first. And this was the first natural site of infection. The index case in Sudan was cotton factory workers. And so these individuals got infected. They were cared for by doctors and by family members, but the infection control wasn't very good, so the infection was spread to other people by contaminated needles among family members, and so that's why this outbreak got so big. Started out in a couple of cotton factory workers and spread to many other people. And it's called Ebola because it's named after a small river in the northwest of the DRC, the Ebola River. Okay, so that was the first one, and then there were a few outbreaks in the next few years, and this is a timeline of the outbreaks. These are the number of cases on the y-axis, and then we have the year on the bottom. You can see the DRC, Sudan outbreaks, and then there were a couple of others subsequently of fewer cases, fewer than the 300 here. The size of the circle tells you how many people were infected. Then there was a long period with no reported Ebola infections anywhere. And then in the late 90s, a series of outbreaks again in DRC, big one in DRC and other surrounding countries, Uganda, another big one. And you can see just a series of outbreaks over the years, never really more than 400 and so cases. And each of these outbreaks was a separate zoonotic transmission from an animal to a human. So this virus, after each outbreak, seemed to disappear. The next one was caused by a new introduction. So this never sufficiently adapted to humans to become a human virus, to be transmitted human to human, but always requires a new introduction from the reservoir into humans. All right, so that's very important. Each of these is a brand new zoonotic outbreak. That's why I call this a dead-end virus, because even though it can spread from person to person, and you see caused 28,000 infections in the most recent outbreak, it can't sustain itself. Eventually, we can stop it if we simply impose infection control that stops transmission. It's never able to adapt to human-human transmission, as is HIV or measles or polio. Most recent outbreak, of course, in Western Africa, 2015, 28,000 plus infections, documented, laboratory documented infections with about a 40% case fatality ratio. That was the biggest to date, obviously. Ebola virus is a BSL-4 pathogen, biosafety level 4. There's BSL 1, 2, 3. So what we do in my lab is BSL 2. BSL 4, I got the opportunity to actually visit the one in Boston, which has been under construction for many years. We went a couple of years ago. It's not yet open, so they let us go in and do a documentary, which we put online. It's called Threading the Needle, where we get to walk through. We could suit up. You have to wear basically a very thick suit, two pairs of gloves, and then you have air pumped into this so that you can breathe. And as you move around the facility, you detach and reattach. These hoses are hanging from ceilings everywhere, so you can move around. And then when it's time to leave, you go through a chemical shower that decontaminates your suit, and then you take it off and you leave the facility. So you have to sign in and sign out, and it's a very, very tightly controlled access and so forth. So to work on Ebola and similar viruses, you have to do so in this kind of facility. And these viruses, Ebola, Marburg, Lassa, have high mortality. They're spread person to person, and we don't have any vaccines or antivirals, and that's part of the reason why they're considered BSL-4. We have about a dozen BSL-4s in the US. The one in Boston is the most recently built, brand new, state-of-the-art. It's amazing. Look at this documentary. You will see how incredibly engineered this place is to be safe. It's basically a concrete cube within a bigger building. And then it has incredible redundancy of systems and so forth. The people in Boston are not happy about this opening, so they have been working for years to try not to get it to open. But I think it's probably the safest place in the world for working on these viruses, and I would have a look at that documentary. Now, Ebola virus, of course, is a negative strand RNA virus. It's filamentous. It was an unusual discovery when it was first seen in the electron microscope to have this filamentous shape. An envelope virus with a nucleic capsid comprised of negative strand RNA with proteins bound to it. And this RNA encodes a number of subgenomic mRNAs that each encode the viral proteins, the RNA polymerase, structural proteins, and look, interferon antagonists, tetherin antagonists, all antagonists of innate responses. And that's probably why this virus can be so lethal. So this is a classic zoonosis. Infection starts in an animal and goes to people. Typically, the index case is contact with animal carcasses, like bushmeat. People go hunting for bushmeat, which could be a bat, it could be a chimp, or stuff you find on the forest floor. Many people will find a dead chimp and bring it home and cook it. And if it has Ebola, that starts the outbreak. Now, we don't always have the index case identified, but in many cases we can see it started with this person who ate bushmeat, and that's where the infection began. And then that's transmitted to other humans by close contact with infected fluids, because these individuals get rapidly infected, they shed a lot of virus, and with close contact, it's easy to spread it. But the chains tend to be short. What I mean by that is that one person will infect maybe a few other people, and then that's it. You don't have very, very long chains of infection as you would for, say, influenza virus. I gave it to you, you give it to another person, and on and on and on and on. And the R0 is 2. That's the number of people on average infected by any one person infected with Ebola virus. So that's kind of low. In terms of where the virus is, Marburg has been found in a fruit bat that is cave dwelling. So virus has actually been found, but as far as Ebola virus goes, only Zaire Ebola virus, one of the 5 different types, only RNA has been found in a bat. They found antibodies in some bats as well, but nobody has isolated infectious virus. So I think it's not clear if this is in fact the host. The antibodies could be simply cross-reactive antibodies. The RNA, it's not the entire genome. So unless you have infectious virus, I think it's not clear if that's the host. But let's assume it is the reservoir of Ebola virus. Then the idea would be these animals are fine with infection and they transmit it to other species which are dead-end hosts. Humans, gorillas, and chimpanzees seem to be dead-end hosts. They get very sick and often die from infections. And so it's thought that, in fact, infection of gorillas with Ebola viruses is helping to wipe out populations of gorillas in many parts of Africa. And of course, if a chimp is infected and dies, and a human then comes and sees this carcass, will take it for bushmeat, and then that starts a new chain of infection. So here's the summary of what I've just told you. You have bats that act as reservoirs. They infect each other with the virus. They spread it amongst each other. They're relatively healthy. They can spread it to dead-end hosts like chimps and gorillas, maybe even some other animals in the forest. We don't know. I mean, you can imagine these are hard experiments to do, because if you have an animal that you think might be infected, you have to take it to a BSL-4 laboratory to do the study. So it's not an easy experiment to do. And then eventually humans get infected, maybe through direct contact with bats. People do catch bats for bushmeat, or from contact with any of these other infected animals as well. So they're dead-end hosts, classic zoonotic infection. Here are 2 examples of some outbreaks. There was one in Gabon in 96 of 37 cases. So a chimp was found dead in the woods. It was eaten by hunters. They just picked it up and brought it back, and they ate it. 18 people involved in butchering became ill, and then 10 other cases in their family members. You have to understand that these individuals will eat the meat, bring it home to their families, and then the infection spreads from there. Gabon, another one, 60 cases. The index case was a hunter who lived in a forest camp, and a dead chimp was found in the forest at the time he got infected. So these are just 2 examples where we know the index cases had some contact with bush meat, but most of the time we do not. Now, the most recent outbreak in Western Africa, Liberia, Sierra Leone, and Guinea, began. So here's the early epidemiological tracing of that outbreak, which I think is really quite informative. So it begins in Milandiu village, and with 9 deaths in December of 2013. And it's thought that this all began with a 2-year-old child who got sick on December 2nd, died 4 days later. He had black stool and vomiting, and that's the index case, S1. And then this child passed on the infection to his sister, his mother, his grandmother, to a nurse, to a village midwife, who then passed it on to another family. And then it traveled to different villages, because some of these individuals provide care to different villages. So you can see it goes to another village here, and to another here, and to another here. They could actually trace all of these cases back to the very start. Now, how this child got infected, we don't know, because there was no history of bushmeat consumption. The child may have been in the woods or near the woods or somewhere which had some contact with infected animals, but they weren't able to get any information before the child died. So that began the outbreak, and down at the bottom here, we're in March 2014. So this went on for 3 months before anyone recognized that it was Ebola, and by then it had spread extensively. And that's why, it's one of the main reasons I think in the end it went so far, because people didn't recognize it was Ebola. It had never occurred in these countries before. No one was used to seeing it, and so it spread extensively, and that's why it was very hard to contain at the end, because this is a tough virus if you don't have good healthcare facilities. As I said, it's spread by contact with infected blood or body fluids. The virus is in urine, saliva, sweat, feces, vomit, breast milk, semen. So if someone is very sick and they're vomiting, you're going to get infected by that if you're taking care of them, especially if it's a family member. If the individual has died and you're preparing the body for burial, you can get infected as well. There's also contact with contaminated objects, needles and syringes that aren't properly sterilized, but also blankets. If someone has been sleeping in a blanket and they're sick and you didn't know it initially, the blanket's contaminated, and if someone else uses it, it can pass on the infection. It's not transmitted by insects, water, food, or aerosol. During this outbreak, there's a lot of discussion about aerosol transmission, and so it can be transmitted by large droplets. Let's say you are a physician and you're intubating someone who has Ebola, right? If you're not properly protected, when you intubate someone, the fluids are going to splash out of them right into your face. Yeah, that's an airborne transmission because you're 3 feet from the patient. But me talking will not transmit Ebola to you. It's not transmitted by aerosol. So the press had a whole tough time with distinguishing between airborne and aerosol transmission. So that's why I have spent some time with you trying to explain the differences. How does it get in you? Mucosal surfaces, upper tract, breaks or abrasions in the skin, contaminated needles. And so because the virus is in so many body fluids, it's very easy for it to infect a new person, especially when they're close by. The incubation period is between 2 and 21 days. Interesting, during this period, you're not infectious. And there are no typical symptoms of Ebola. So you wouldn't know that someone is infected at that time. But then as soon as the outward symptoms occur, rash, hemorrhage, convulsions, and so forth, then you become contagious. And as I said, the case fatality rate is 30 to 90%. And so there was also a big discussion in the press about should people be allowed to come back from working in West Africa, when they may be incubating Ebola. And as I told many people, there's no evidence that anyone who has not got these severe symptoms is infectious. And there was a ER doctor from Colombia who came back from working in West Africa, and he was here in New York City. He went bowling on a Friday night in Brooklyn, and the next day he developed a fever. He was hospitalized, and they said, well, he was probably contagious that night. No, he wasn't, because he didn't have any of the symptoms of infection. And they wanted to quarantine people who had worked in these areas not having any symptoms, which is absurd. It's not consistent with the biology of infection. Anyway, all that is over, but presumably it'll return at the next outbreak. This is a very serious illness, multi-system involvement. Systemic infection, prostration, gastrointestinal involvement, respiratory involvement, involvement of the circulatory system, involvement of the neurological system. The virus seems to have a very broad tropism and can replicate in all of these different places. It causes extensive necrosis in many different organs. It infects a large number of cell types, as you can see here. It causes liver damage and massive lymphocyte death, even though the lymphocytes don't seem to be infected. So clearly, infection is causing release of lots of cytokines, which in themselves are causing a lot of these systemic syndromes. Lots of inflammatory mediators, especially by infection of macrophages, imbalanced cytokine production, and coagulation systems become impaired. And that's often why you have the extensive hemorrhagic, the bleeding associated with infection. So I always teach the Ebola virus as an example of an acute infectious disease. But many weeks after some of these individuals who were infected recovered, virus was found in ocular fluid, 9 weeks after clearance of viremia, which of course is virus in the blood. And there's some damage in the retina here, you can see, caused by infection. So there's virus in the vitreous humor, which is an immunoprivileged site. How it got there and how long it was there is not known, because there are very few cases to study, so we're not really sure. There's also some evidence for sexual transmission because the viral genome has been observed in semen. There were some early suggestions epidemiologically of sexual transmission, and this study was done in 93 men who were in the area of the outbreak and may have been infected, and they took semen samples from all of them, and 46 of them tested positive for Ebola virus RNA. So these are all confirmed infections, and you can see months since onset of infection. You can see viral RNA detected in semen for extended periods of time. So we don't know, again, if this is infectious, because these are PCR assays. We're just looking for RNA fragments. Whether or not it's infectious and transmissible isn't known. So what did we learn? Every infectious disease is a global problem. You know, the previous Ebola outbreaks were in Africa. Nobody really cared too much. But as soon as they came to other countries, here in the US, we had 2 imported cases, one into Dallas and one into New York City. And then the case in Dallas spread infection to 2 nurses, so 2 locally acquired infections. The New York City case was the ER doc that came in and developed infection here. And the Dallas case, again, came in healthy apparently and developed the infection here, although they were acquired overseas. So that made for a lot of excitement. We do have Ebola virus vaccines. They've actually been ready for clinical trials for a long time, but they were never tested because there was no money to pay for them. But as soon as this outbreak started, they were put into testing, and they got to a certain point where they looked pretty good, but then the outbreak stopped. And that always raises the question is, what other viruses should we be preparing for? You can't make a vaccine for every virus that is out there. So that's an example of a brand new emerging infection and the factors that lead to its emergence. Let's end up talking a little bit more about Zika virus, which we have touched on several occasions in this course. First identified in 1947 in Uganda, all right? It was isolated from this Aedes species of mosquito, and then first isolated from people in Nigeria in 1954. For the next 50 years, it was detected throughout Africa and Asia by serology mainly. Appears to be spread by Aedes aegypti, but only 20 cases or less over the next 50 years. So not a real attention getter in terms of emerging infections. So that's summarized on this graph. This is a lovely graph with colors that indicate the earliest evidence of Zika circulation by virology or by serology. So you can see the colors correspond to when. You can see that the virus was detected. You can see the virus is slowly moving from Africa, throughout Africa, moves into Asia and so forth. And then, of course, nothing serious for 50 years, but in 2007, there was an outbreak on Yap Island, which is right here, the first large outbreak. So we'd never seen any outbreaks, just like polio. And then all of a sudden, boom, we have large outbreaks, 8,000, 73% of the population. And then it spread to other islands in the Pacific as well, causing outbreaks. People started to pay attention. And then, of course, when it reached Brazil in 2015, caused a huge outbreak, which was then associated with birth defects. And that's when the world started to pay attention to this. This is, of course, a Flavivirus. It's an envelope virus with a plus-stranded RNA genome that encodes a polyprotein. Makes one long protein that's processed by viral and cellular proteases. And here's a lovely picture that I put together. Within 3 months, the cryo-EM structure of this virus had been solved, and the coordinates allow me to make this picture. This is the surface. Remember, inside there's the RNA, then there's a capsid made of protein. Then around it there's a membrane, and embedded in the membrane is the E-glycoprotein, which is a type 2 fusion protein. So it's laying flat on the virus surface, maybe made up mostly of beta strands. And you can see how it's icosahedrally ordered. Here's a 5-fold axis of symmetry, a 3-fold, and a 2-fold. Really clear from this, something we talked about years ago. So even though it's an envelope virus, the proteins are icosahedrally ordered because of the way they interact with each other. This virus causes a mild rash-like disease, joint pain, conjunctivitis, headache, very similar to dengue and chikungunya, with a short incubation period. Only 1 in 5 people develop symptoms. The other 4, they're infected, but no symptoms of infection. And up until recently, no fatalities caused by this virus. It is shed in a number of fluids: semen, urine, saliva, blood, breast milk. We don't know how virus gets to these sites. We don't know the risk for shedding at these sites. In addition, we know that mosquitoes, of course, pick up the virus from blood. We don't know if there's any transmission via saliva or urine. We believe there is sexual transmission by semen, but it's probably very, very low frequency. The main mode of transmission is via the mosquito. The CDC surveillance report, which was issued at the end of last year, they looked at 2,300 cases of infection, which were imported. 23 out of the 2,300 resulted from sexual contact with a traveler to an affected area. So it's about 1% risk of sexual transmission. The risk of birth defects, about 10%, but that's probably an underestimate. So CDC advises pregnant women not to go to endemic area or don't have unprotected sex with people who have been to these endemic infected areas. RNA seems to persist in semen a long time, but no one has looked for infectious virus. They've looked for fragments of RNA. And in a mouse model, in fact, you can find Zika RNA in semen for 50, 60 days, but infectious virus stops at day 20. So I'm not sure that this 188 days is all infectious virus in people. That's obviously important. The key point here is that this virus cannot be sustained by sexual transmission alone. If you took away all the mosquitoes, it would not spread globally by transmission because the efficiency of transmission isn't good enough. So what I call this is a sexually transmissible infection, but it's not sexually transmitted. Sexually transmitted would be a virus like HIV, where it's one of the main means of transmission, as we'll see next time. So far in the US, we have had 5,000 travel-associated cases in these colored states. Most of the states have had at least one travel-associated infection. And again, coming from other areas, being infected, bringing it back here and getting diagnosed here. So 5,000. 223 locally vector-acquired cases, 217 in Florida and 6 in Texas. So these are people who got infected while in the US. So not a lot. And the US territories, huge numbers. Puerto Rico, US Virgin Islands, Samoa, 36,000 cases, most of those in Puerto Rico. They've been hit very hard with infection and subsequently had lots of microcephaly. Now, the vector for this virus is Aedes aegypti, and its range is shown on this map. So if you can see this kind of shaded area here, which comes from the southwest through Texas, the southeast, and then up the eastern seaboard. That's the range. That's the known maximum range of Aedes aegypti. In reality, it goes all the way up to New York, but we don't have any Aedes aegypti here. We haven't had any for about 50 years or so. Aedes albopictus is another species which we've talked about, the invasive mosquito, in which the virus can replicate, but it's never shown to be transmitted in it. People are worried that it might be transmitted by albopictus. It has a more northern range. That's the dotted line here. And all these circles are areas where these two mosquitoes would be abundant at some time of year, and those are the import areas where people travel into the country. And the two areas where we've had local transmission, which means you need to have Aedes aegypti and people who are infected. One is in the Miami area, where we know there are Aedes aegypti, and as I said, there were a number of cases locally transmitted there, but those died out relatively quickly. And then Brownsville, Texas, the most southern part of Texas here, where we also have Aedes aegypti. And it's not surprising because there have also been dengue outbreaks in these areas, which is again vectored by Aedes aegypti. So not surprising. The rest of the country, these are not threats for transmission because people aren't outside that much. They have air conditioning. They have screens on their windows. They have a very different lifestyle that does not encourage vector biting. So we've had minimal local spread in the US, nothing in albopictus. And just for a comparison, back in 1793, there was a yellow fever epidemic which reached as high as Philadelphia because the mosquitoes had gone up that far. But mosquito control, the use of insecticides, screens, and air conditioning and so forth got rid of the mosquitoes, so they're now not present any longer except in that part of Florida. And now you may say, well, West Nile spread across the US in 1999. Well, that was very different. It's a different mosquito with different biting habits, and there is a bird reservoir for West Nile. So those two differences probably help. Culex species are throughout the US, whereas Aedes, of course, is restricted. Now, there are CNS complications associated with infection in both adults and infants, including a variety of rare CNS afflictions, but of course in infants a number of congenital defects which have led to the attention for this virus. So the question is, why is this virus suddenly doing these things when it was benign for so many years? Well, if you look at the sequences of African and Asian isolates of Zika virus, you can see they can be grouped into two parts of this phylogenetic tree. So they are distinct. So as the virus has moved through different populations, it has changed. But has that been responsible for this change in disease? So is it evolution or introduction into a new population? So there's a very nice paper published recently in MBio, Emergence of Zika, Transmission and Congenital Zika Syndrome are recently evolved traits to blame. And they have two theories. One, adaptive evolution, genetic changes in the virus allowed higher replication in vectors, altered tropism to include congenital infection, or stochastic introduction. The virus was simply randomly transported to locations with large enough naive human populations accompanied by large urban mosquito vectors for an explosive outbreak. Formerly rare conditions were suddenly recognized when large enough numbers of infections occurred. And that's completely plausible. That's what happened, I think, with West Nile virus. Now, back in April 2016, Peter Hotez, a well-respected parasitologist, wrote in an op-ed, there are many theories for Zika's rise, but the most plausible is that the virus mutated from an African to a pandemic strain a decade or more ago and then spread east. I don't know why it's most plausible. There's no evidence for it. I mean, there is genetic difference between the African and the Asian strains, but that certainly doesn't prove that the differences are responsible for this change in disease pattern. Equally likely is the stochastic introduction theory. Why didn't he mention it? I don't know. You'd have to ask Peter. He either didn't like it or he thinks the mutation theory is more likely to be bought by the New York Times. But this is really what bothers me when people write for the public about science. This isn't fair because there's no evidence that the mutations have had anything to do with this. All right, let me end up with how common are host range jumps of viruses. They're very common, but most of the time they don't do anything. We only see the ones that do something because they make a big deal, they make disease that's different, and we pay attention to them. Can we predict them? We can't. If anyone says they're going to work on ways to predict the next outbreak, don't believe them. All we can do is be ready. We can have preparedness. We can have ways to react. We can say, here's what we're going to mobilize, here's what we're going to do. We're going to put these treatment centers here and so forth, the way we did with Ebola in West Africa. We can look at what's out there. We could determine the viruses in a variety of species and study them and be ready. But you can never predict what the next one is going to be. It's a random process. But studying what is out there and being prepared, I think, is the best way to deal with it.